Big news for the GPCR community. Discovery on Target is happening September 22nd to 25th in Boston, Massachusetts. Hello, everyone. Yamina here. Welcome to the Dr. GPCR ecosystem. We're partnering this year with DOT to spotlight their exciting program, and in today's video, you'll meet one of the incredible presenters. You'll find all the details and links in the description. I'll be there in person chairing a session, and you won't want to miss Dr. Kenakin's talk on the kinetics of allostery and his short course on the 4 most common causes of drug candidate failure. And remember, Terry's Corner is always available with on-demand pharmacology courses you can access anytime. Plus, GPCR Happy Hour is happening on September 24th. Your chance to connect with colleagues, share ideas, and catch up with friends in a relaxed setting. Find all the links in the description. And now, let's dive in. Hello, everyone. This is Yamina from Dr. GPCR. And this month, we're getting ready for the Discovery on Target GPCR track meeting. And in our collaboration with the DOT team, we have been doing spotlight speaker conversations. And today, I'm very excited to have with me Dr. Terry Kenakin. Terry, welcome.
Thank you. Happy to be here.
So excited to see you today, but also to see you in person in Boston, September 23 to 25.
Fun.
I think it's going to be fun. I don't think we need to introduce you. I know that you're going to be giving a talk on September 24th that I have the privilege and honor to chair in that first session. And I'd love to hear more about what is it that you're going to be talking to us about during that talk. I'm going to let you share your screen.
Yes, I'll be presenting a talk, and I'll be also presenting a course, but I'd like to tell you about some of the new work we've been doing, and it's to do with kinetics. I've been working with George Vaquelin, and I've learned a lot about this. So our presentation will be on what's so special about allostery, what's so special about kinetics, and then we'll put those together, what's so special about kinetics and allostery. So we all know that we get our allosteric constants for compounds. We fit to an equilibrium model, which is the standard way to do this. However, sometimes this falls short because drugs don't really act in an equilibrium world. They act in vivo. Real-time matters, and so the action may be here in non-equilibrium systems, and we have numbers that are describing what we'd see here. So with kinetics, we can sort of get an inroad into this non-equilibrium road. So let's say, see what happens when we look at the allosteric model, which is here, which is a target binding to ligands A and B to form complexes, and we can get constants like alpha. Alpha is the change in affinities of these when the other compounds on board. However, we can redescribe this model in something called fluxes. So these are flux equations which in real time show the changes in the quantities of these various species. Why would we want to do this? Because we can get 3 values of alpha that are constant in 3 different ways kinetically, case 1, 2, and 3. And again, alpha is still constant, but if we dig down, look at the kinetics, we can separate compounds here that will have long residence times and compounds that have the unique property to seek and destroy agonist-bound receptors. So kinetics can give you an added insight, and that's the message of my talk.
That is wonderful. I think people are going to love, I will be there, and I'm going to love hearing more about it. For those who have registered to your day-and-a-half length course, I'd love it if you can give us a little bit of an overview of what the attendees will be learning from you during a day and a half they're going to be spending with you.
Oh, yeah, sure. This one I'm excited about. It starts with a depressing fact, depending on who you read. 50 to 80% of new drugs that come out fail because of efficacy. Think about that. Not safety or all the other issues. These drugs don't work. So we had a team of maybe 30, 40 people working 5 years with resources, doing all they knew what to do to make a compound, put it in humans, and it fails. So that's not a very good success rate. Much has been written about this, and this course is about that. How can we reduce this rather high rate of attrition? And the ideas that are out there are shown here. There's 4 of them. The first is, what efficacy did we need? This will be a question we'll always have. You always have that question. But there's been great progress made in the other 3 reasons. Did we choose the wrong target? Did we choose the wrong chemical? And maybe when we get a compound, did we know everything about it? Did we know its true profile? And this is the one we will concentrate on, although we will cover all 4 of these in the course.
Thank you, Terry. And my last question to you is, considering you mentioned, being excited about things, what are some of the advances in the field over the past few years that you think are very exciting? And if you can expand on that a little bit.
Right. Thank you, because there have been tremendous things happening. To explain those, let me start with the state of the art. When Kenakin was a student, it was fairly simple. You would have an assay and you'd get a signal, most often calcium. Call this the Stone Age of GPCR research. But even then we knew, and with increasing technology, we started to get these new assays. We know a whole lot more is happening, and that's the key. We now know that receptors, I call them physiology's Swiss Army knife. They can modify cell function a number of ways: G proteins, beta-arrestin, MAP kinases, even moving the receptor in and out of cells. So this really opens up the canvas for what we call efficacy. This has been the exciting reveal of the recent years in the field, where we now know the drugs give fingerprints of signaling efficacies to cells. And so we don't just talk about a drug that elevates cyclic AMP, etc. They're doing a whole lot more, and you can almost make a case for every drug has a unique fingerprint, greatly opening up the potential for new therapies. So that has been exciting.
And it goes back to the last point you were making when you were talking about your course, about very well characterizing that molecule before moving it forward. And now we have the tools to do that.
Yep.
So for those of you who have not seen us on LinkedIn, who don't necessarily follow Dr. GPCR, early this summer, we decided to put together this labor of love, which ended up being Terry's Corner, where you can get bite-sized on-demand pharmacology courses on a weekly basis. It's based on Terry's 40 years of experience, his 250 papers and books, all in one place on demand for every pharmacologist and at different levels. So every week you get one new course on specific topics. And I'm excited that this month we're starting off with our monthly Ask Me Anything sessions, where people can come in and ask Terry the tough questions and help answer them. And for any Dr. GPCR Premium members, you all get an early bird discount coupon code that you can claim in your Dr. GPCR weekly news. And Terry, maybe you can tell us a little bit more about the content that's already available.
Yeah, sure. Throughout the years of discovery, we've all encountered issues in research, and it's useful to solve these issues. So I've collected the myriad of issues that I've encountered, and so we give a short course. The video is about 20 slides, 10 minutes or so, on specific topics. And here's what we have in hand already. And we add one each week. And of course, you all can suggest new ones, and we will add them in the order of preference. But And so we also have this.
Yes, can you,
I'll
if you
hand
can
it
go
over
back,
to you here.
thank you, Terry. Can you go back one slide, please? Because what I want to also emphasize is, although this is what's in the collection right now, there is 52 other courses coming up. Terry's productive nature is unleashed in Terry's Corner, and the courses are for different levels. You have foundational, intermediate, and drug expert drug hunter courses, and for every course, you get the course video, you get the key insights, takeaways, and Terry's recommendation for papers to read. So it's one course, one bite-sized pharmacology piece directly from Terry himself. And Terry, now we can move on to the next slide.
Okay, thank you.
And if you want to get a little bit of a sense of what comes out every week, we have created the Kenakin Brief, our weekly newsletter, where every week delivered into your inbox are the latest insights on the last course, and you get a chance to get a taste of what's waiting for you in Terry's Corner.
All right.
And with this, Terry, I'm excited to see you in Boston in September, and I'm glad that you'll be able to join us for the GPCR happy hour.
I am too, yes. Great.
Wonderful. I'll see you in Boston very shortly. Thank you so much, Terry. Don't go anywhere.
Okay, folks. Bye.
I can't wait to see you in Boston for DOT 2025, especially at the GPCR happy hour on September 24th, where we'll connect, exchange ideas, and unwind together. And don't forget, Terry's Corner is ready when you are, with on-demand pharmacology courses. Check the links below for all the details. Until next time, this is Yamina, signing off.